They tested 1,000-year-old mummies in Peru for tuberculosis. The disease was already there. Peru, 1,000 years before Columbus, before any European ship crossed the Atlantic. Today, tuberculosis kills over 1 million people every year. It remains the deadliest infectious disease on Earth, more deadly than HIV. And its origin story was wrong from the start. In Peru, TB is still a public health emergency, and the assumption was always that Europeans brought it. The bones showed the unmistakable signature of tuberculosis. Lesions on the ribs, damage to the spine. For decades, the assumption was simple. Europeans brought tuberculosis to the Americas. 1,500 years of history compressed into a single narrative. Contact equals disease, Europeans equal source. The narrative was taught in every medical school, every public health program, every history textbook. Cortez brought measles, and somebody brought TB. That was the story. But the mummies were 1,000 years old, 5 centuries before any European reached South America. The civilizations that lived here were sophisticated. Moshe Chimu, Wari. They built cities and irrigation systems. Some of their ceramics show faces with deformities. Paleopathologists have debated whether these depict TB symptoms. The Moshe documented their world in clay disease surgery. Death. If TB was present, they may have depicted it. The tuberculosis was already killing people in Peru, while Europe was still in the early medieval period. The question was not whether TB existed in the pre-Columbian Americas. The bones proved it did. The question was where it came from. Pott's disease, spinal tuberculosis. The vertebrae collapse and fuse. The evidence in ancient bone is unmistakable. The lesions in the Peruvian mummies matched the clinical pattern precisely. This was not arthritis, not trauma. It was tuberculosis. Paleopathologists had seen these lesions for decades. They knew it was TB, but not its source. Over 200 documented cases of pre-Columbian TB across the Americas, all before ancient DNA could explain the source. If Europeans did not bring it, then how did tuberculosis reach Peru 1,000 years ago? In 2014, Kirsten Bose at the University of Tübingen extracted ancient pathogen DNA from the remains. They sequenced 3 complete TB genomes from the ancient Peruvian bones, and the results overturned everything scientists thought they knew about tuberculosis in the Americas. Ancient pathogen DNA is even harder to recover than ancient human DNA. The bacterial genomes are tiny compared to the human genome. They used targeted enrichment, fishing for TB DNA in a sea of human and environmental contamination. The baits were designed to match known TB genome sequences. They pulled bacterial DNA out of a mixture that was 99% human and environmental. The bacterial genomes were fragmented but complete. The ancient TB could be placed on the evolutionary tree. 3 ancient genomes from a Peruvian desert. Enough to overturn a century of assumptions about disease origins. The TB strains from Peru did not match any old world human tuberculosis. Not a single one. They matched something else entirely. Something nobody had predicted. Seals. The ancient Peruvian TB strains were most closely related to tuberculosis found in marine mammals. Mycobacterium pinnipedii, a strain of tuberculosis that circulates in seal and sea lion populations across the world's oceans. Sea lions and fur seals gather in massive colonies along the Pacific coast. Thousands in close contact. TB spreads easily in these dense colonies. Respiratory transmission, contact with infected fluids. The bacteria thrives in crowded conditions. A coughing seal on a beach can transmit TB to any animal within a few meters, including a human hunter. The bacteria is extraordinarily hardy. It survives in dried tissue for weeks, in soil for months, in bone for millennia. Under UV light, the bacterial DNA hotspots glow. 1,000 years later, the chemical footprint remains. The waxy cell wall of TB bacteria is one of the thickest in nature. It protects the DNA inside even after the cell dies. A sick seal on a beach. A hunter butchering the carcass. One breath. That is all it takes. Tuberculosis jumped from seals to humans on the coast of South America, centuries before Columbus. Coastal South Americans lived alongside marine mammals. They hunted seals, processed seal meat, and blubber. Every interaction was an opportunity for zoonotic transfer. Butchering a sick seal, breathing in bacteria. Archaeological sites along the Peruvian coast are filled with marine mammal bones. Seal hunting was not occasional. It was systematic. The midden layers are meters thick, centuries of accumulated seal bones. This was an industry, not a supplement. They depended on seals the way inland cultures depended on maize. It was the foundation of their economy. The butchering process exposed hunters to every fluid in the animal's body: blood, lung tissue, respiratory secretions. The seal TB strains diverged from other marine mammal lineages between 700 and 1000 CE. The timing matched the archaeological evidence. The mummies with TB lesions dated to the same period. Two independent dating methods: molecular clock from the bacterial DNA, radiocarbon from the bone, both pointing to the same centuries. When the genetics and the archaeology agree independently, the case is closed. But how did seal TB cross the ocean? South American and African seals carry the same TB lineage. Seals do not respect continental boundaries. They cross oceans, and they carry their diseases with them. African fur seals and South American sea lions share the same TB lineage. It crossed the Atlantic inside them. Marine mammals migrate vast distances. Southern elephant seals cross between Africa and South America routinely. Every migrating seal that carried TB was a potential vector. The ocean did not contain the disease, it spread it. Individual seals have been tracked traveling over 5,000 kilometers across the Atlantic. One infected animal can carry TB to a new continent. They cross oceans the way birds cross continents. The Atlantic is not an obstacle for a southern elephant seal, 1/6 seal. One ocean crossing, one beach in Peru. That is how a disease jumps continents. We think of oceans as barriers. For marine mammals, they are highways. Their diseases travel with them. Once the bacteria reached South American marine mammals, the jump to humans was a matter of proximity and time. The respiratory route is the most efficient. TB bacteria from a cough can remain airborne for hours. Butchering was the highest risk activity. Direct contact with infected lungs. The bacteria aerosolized during the cutting. Now here is where the story gets bigger. The seal origin of American TB was surprising enough, but the Boas study also challenged the origin story of TB itself. For decades, scientists believed humans caught tuberculosis from cattle, the bovine origin theory. And it was wrong. The textbook story said TB evolved in cattle and jumped to humans during Neolithic domestication 10,000 years ago. Genomic studies by IT Comas had already begun dismantling this theory before the Peruvian finding. The logic seemed airtight. Cattle carried TB humans, domesticated cattle 10,000 years ago. Therefore, cattle gave TB to humans, but the molecular clock said otherwise. Human TB lineages diverged from each other long before cattle were domesticated. The oldest human TB lineages diverged from each other roughly 70,000 years ago. Cattle were domesticated 10,000 years ago. The math does not work. The bovine origin theory survived for decades because nobody sequenced enough ancient TB genomes to disprove it. The molecular clock data showed that human TB strains are older than bovine TB strains. Not younger, older. The Bayesian analysis was conclusive. Human TB lineages coalesced tens of thousands of years before the oldest bovine strains. Comas and colleagues published the finding in Nature Genetics. The bovine origin myth was dead. Tuberculosis did not jump from cattle to humans. It jumped from humans to cattle, backwards. Humans gave TB to their livestock, not the other way around. Mycobacterium bovis, the cattle strain, descended from the human lineage. Cattle caught it from us after domestication. And in the Americas, the story was different again. TB arrived not from humans or cattle, but from seals. TB originated in humans in Africa. Over 70,000 years ago, it migrated out of Africa inside its human hosts. Older than agriculture, older than civilization. It has been with us from the beginning. TB co-evolved with humans. It adapted to our immune system for tens of thousands of years, 70,000 years of coevolution. TB knows the human immune system better than almost any other pathogen. The Peruvian TB genomes revealed another critical detail. The seal-derived strains were eventually replaced. When Europeans arrived, they brought their own TB strains, human-adapted strains that were far more virulent. The European strains outcompeted the seal-derived strains. Within centuries of contact, the original American TB lineage was replaced. Today, all tuberculosis in the Americas belongs to European-derived lineages. The seal strain is extinct in human populations. The European strains had tens of thousands of years of coevolution with human immune systems. The seal strains had centuries at most, in a direct competition between strains inside a human host. The human-adapted European strains won every time. The indigenous peoples of the Americas were hit by TB twice. First from seals, then from Europeans, two separate origins for the same disease. The second one, combined with every other European disease, devastated them. The replacement happened within generations of contact. By the 17th century, the seal-derived lineage was functionally extinct in human populations. The indigenous population was hit by two epidemics of the same disease from two completely different sources. The indigenous population was now fighting a disease it had some resistance to, from centuries of seal TB exposure, plus a new, more aggressive strain it had never encountered. The implications of the seal origin theory extend beyond history. They reshape how scientists understand zoonotic disease transfer. Tuberculosis is not a single species disease. It moves between species more freely than anyone expected. Modern marine mammals still carry TB: sea lions in California, fur seals in Australia. The reservoir is active. The same zoonotic pathway from 1,000 years ago is still open. The bacteria persists in seals. People who work with marine mammals today, veterinarians, researchers, rehabilitation workers, face the same exposure risk that ancient Peruvian hunters did. The study also forced a rethinking of TB evidence found elsewhere in the pre-Columbian Americas. TB jumps species more often than any other major pathogen. The Mycobacterium tuberculosis complex is a generalist. In Britain, TB transmission between badgers and cattle remains a major problem. The same cross-species dynamics. The pattern was clear once researchers looked for it. Pre-Columbian TB cases clustered along coastlines, not inland. Coastal populations got TB from seals. Inland populations rarely showed the disease before European contact. The geographic distribution matched the zoonotic pathway perfectly. Seal contact required coastline. TB followed the coast. The coastal clustering was key. If TB came via Beringia, it should appear inland first. It did not. Highland Andean populations showed almost no TB before European contact. The disease was coastal, marine, from the sea. The geography of the disease matched the geography of seal hunting. That is not a coincidence. Previous researchers had attributed all pre-Columbian TB to either early human migration from Africa or independent evolution. The seal pathway had never been considered. The Boss study settled the debate for South America. The genomes were unambiguous. Seal TB, not human migration. 3 hypotheses tested, only one survived. The marine mammal pathway was the only one consistent with the genomic evidence. The academic debate effectively ended with the 2014 publication. The genomes were too clear to argue with, not people walking across Beringia. The transmission vector was marine mammals. The broader lesson from the Peruvian TB study is about assumptions. Scientists assumed one pathway because it seemed obvious. The study reshaped the entire field of paleopathology. Ancient pathogen DNA became a primary tool for understanding disease history. Before Boas, most ancient disease diagnosis relied on skeletal lesions alone. After Boas, genomic diagnosis became the gold standard. The combination of skeletal analysis and genomic confirmation eliminated guesswork. Every ancient disease diagnosis could now be verified at the molecular level. The field of ancient pathogen genomics exploded after 2014. Plague, leprosy, smallpox, all sequenced from ancient remains. Over 500 citations in the first decade. The seal origin of American TB became one of the defining findings of ancient genomics. Europeans brought diseases to the Americas. That is true, but not all diseases arrived the same way. Smallpox came from humans. Measles came from humans, but tuberculosis came from the sea. Every other major disease came from human contact. Tuberculosis was the exception. The ocean delivered it through an animal intermediary. The relationship between coastal people and marine mammals continues today, and so does the zoonotic risk. We mapped every disease to its human origin. We missed the one that came from the ocean. The coast that gave Peru its first epidemic. The animals that carried it, the ocean that connected them, all still here. The ocean was not a barrier. It was a highway for people, for animals, and for their diseases. The mummies kept the evidence sealed for 1,000 years. The bones recorded the infection. The DNA identified the source. They tested Peruvian mummies for tuberculosis. The disease did not come from Europe. It came from the sea. 1,000 years buried in the desert. The mummies kept the bacteria sealed inside their bones. The dry desert air mummified the bodies, and inside the bones, the bacterial DNA survived intact for a millennium. Desert burial The perfect preservation environment. The mummies were a time capsule, and the TB DNA was the message inside. The same desert that preserved the mummies preserved their pathogens. A 1,000-year-old crime scene with the evidence intact. The desert kept the secret. The lab read it. And the SEALs told the story nobody expected. SEALs carried it across the Atlantic. Hunters caught it on the shore. The DNA proved it. The disease was already here, long before any European arrived. And it came from a source nobody expected. It was already here. The mummies proved it. The DNA explained it, and the SEALs carried it.